Welcome to everyone present here. Today let's see about mass spectrometry. Mass spectrometry is a powerful analytical technique used to quantify the known materials as well as to identify the unknown materials with a sample. It is used to elucidate the structure and chemical properties of different molecules. It is a complete process which involves the conversion of sample into gaseous ions with or without fragmentation, which are then characterized by their mass to charge ratio. This technique basically studies the effect of ionizing energy on molecules. It depends upon chemical reactions in the gas phase in which samples are consumed during the formation of ionic and neutral species. Then let's see about the working principles of mass spectrometry. The sample may be solid, liquid or gas, which is ionized by bombarding with its electrons. This may cause some of the sample molecules to break into charged fragments. These ions are then separated according to their mass to charge ratio by accelerating them and subjecting them to an electric or magnetic field. Ions of the same mass to charge ratio will undergo the same amount of deflection. The ions are detected by a mechanism capable of detecting charged particles such as an electron multiplier. It results in the displayed as a spectra of relative abundance of detected ions as a function of mass to charge ratio. The atoms or molecules in the samples can be identified by correlating known masses to identifying masses or through characteristic fragmentation pattern. Then let's see the components present in the mass spectrometry. This instrument consists of 5 essential components such as inlet, ion source, mass analyzer, detector and recorder. Inlet introduces the samples from the ambient room temperature into ion source. Ion source converts the sample molecules into ions. Mass analyzer helps to separating ions according to their mass. Detector helps to detect ions and amplifying the signal. Recorder receives the signal from the detector, further amplifying, recording and creating a mass spectrum. This is a simple flowchart for mass spectrometry. First, the sample is introduced into ion source, and the ion source then converts into mass analyzer. Mass analyzer helps to separate ions according to their ions, and it leads to detector. Detector helps to detect the ions and it leads to the signal. With all the above components, a mass spectrometer should always perform the following processes. First, produce ions from the sample in the ionization source. Then separate these ions according to their mass to charge ratio in the mass analyzer. Eventually, fragment the selected ions and analyze the fragments in a second analyzer. Then detect the ions emerging from the last analyzer and measure their abundance with the detector that converts the ions into electrical signals. Then process the signal from the detector to the transmitter to the computer and control the instrument using a feedback. These are the steps and instrumentations of mass spectrometry. A sample is introduced into an outlet, then gas phase. With the help of gas phase, it gets into the source, then it gets into the analyzer, and then it gets into the detector. Then detector leads to signal the data. Sample inlet. A sample stored in the large reservoir from which molecules reach the ionization chamber at low pressure in a steady steam by pinhole called molecularly. Ionization. The atoms are ionized by knocking one or more electrons off to give positive ions by bombardment with a stream of electrons. Most of the positive ions formed will carry a charge of +1. Ionization can be achieved by electron ionization, chemical ionization, disruption technique. Deflection. Ions are deflected by a magnetic field due to differences in their masses. The lighter the mass, the more they are deflected. It also depends upon the number of positive charge ions carrying. The more positive charge, the more it will be deflected. Then detection. The beam of ions passing through the mass analyzer is detected by a detector on the basis of mass to charge ratio. When an ion hits the metal box, the charge is neutralized by an electron jumping from the metal onto the ion. Then let's see about the several applications of mass spectrometry. Mass spectrometry is applied in environmental monitoring and analysis. For example, soil, water and air pollutants and water quality, etc. It applied in geochemistry for determining age determination, soil and rock composition, oil and gas surveying. Chemical and petrochemical industry helps to control quality. Identify structures of biomolecules such as carbohydrate nucleic acids. It is applied in sequence biopolymers such as proteins and oligosaccharides. Determination of molecular mass of peptides, proteins and oligonucleotides. Monitoring gases in patients' breath during surgery. Identification of drug abuse and metabolites of drug of abuse in blood, urine and saliva. The analysis of aerosol particles. Determination of pesticide residues in food.